Steps in muscular contraction. The first step begins when the nervous system generates nerve impulses that signal to conduct an action potential that arrives at the end of the neuromuscular junction. Acetylcholine is released from axon terminals, which then binds to active site receptors on motor end plates, which opens them, and the receptor protein channel opens and increases permeability of the sodium ions into the sarcoplasmic reticulum. Action potential will occur. Sodium ions then enter muscle fibers. Rapid depolarization of sarcolemma occurs, and the action potential into the T tubules. This voltage change, it goes to a less negative charge. The action potential spreads away from the end plate in all directions and depolarizes the T tubules. The steps 5 through 10 are part of the latent period, which is the lag time between stimulation and contraction. The action potential continues down the T tubules into the sarcoplasm, where it depolarizes and releases of calcium ions, and it goes into the sarcoplasmic reticulum membrane. The SR responds to the action potential by opening up the calcium ion release channels, which floods the surrounding sarcoplasm located between the thick and thin filaments. Therefore, calcium ions combines with troponin and diffuses into the muscle fibers, and a contraction begins. Basically, to break it down, the action potential travels to the muscle fiber, the steps occur in the muscle fiber that results in a contraction, and the sarcomeres contain thick filaments, which is myosin, and thin filaments, which is actin, and both eventually create a cross bridge together. Troponin changes shape and exposes the myosin head binding sites on the actin when calcium ions bind to troponin. Myosin heads, which are known as the cross bridge, attaches to actin binding sites on the thin filaments. ADP and phosphate powers the movement of the myosin head. Myosin head pivots and it tilts, otherwise known, and brings the actin filaments of sarcomere toward each other and pulling actin filaments. This is also called the power stroke. Once myosin head is pivoted, the ATP binding site is exposed and ATP binds to the head. The myosin head detaches, releases from the actin binding site. The myosin head then moves to the cocked forward position. Myosin head then attaches and re-extends into a ready position onto a new binding site on actin. Contractions are repeated over and over as long as the ATP and calcium are available. Calcium is returned to the SR by calcium ion active transport pump. The sarcoplasm now has less calcium. Troponin again covers the actin binding sites, and there is no myosin-actin interaction can occur. Muscle fiber then relaxes. The movement of relaxation is due to the elastic effect or due to the connective tissue in the muscle. Important to remember, ATP is required for 3 major roles in contraction: the reposition of the myosin heads, detachment of the myosin head from actin once the power stroke is complete, and the power of the calcium ion active transport pump that rapidly removes the calcium from the sarcoplasm back into the sarcoplasmic reticulum, which is known as the reservoir. Thank you for viewing.